everybody. We're just going to give maybe another minute or two to let people join the meeting, and then we'll get started. Hope you're all having a lovely morning. And feel free to introduce yourselves in the chat as well. Maybe a good chance to get to know each other, get connected. Give it one more minute. All right, great. Let's get started, and I'm sure we'll get more people joining as well. But hello, everybody. Welcome to our June edition of the Science of Alt Protein APAC edition seminar. And my name is Arun Naidu, the SciTech Engagement Specialist at GFI APAC, and I'll be your host for today. It gives me great pleasure to welcome Dr. Nazneen Sofio as our speaker to talk to us about altering products of lipid biosynthesis pathways through enzyme engineering and precision fermentation. Dr. Nazneen Sofio is a renowned synthetic biologist and senior research fellow from the Singapore Institute of Food and Biotechnology Innovation, SIFBI, part of ASTAR, the Agency of Science, Technology, and Research. Dr. Nazneen Sofio also happens to be a GFI grantee as well. With an engineering degree in biotechnology and doctoral training in biochemistry and molecular biology, Dr. Nazneen Sofio is interested in synthetic biology, protein characterization, metabolic and protein engineering, protein structure-function relationship, protein assays, gene regulation studies, and bioinformatic analysis. Through her work, she has built an extensive experience in genetic manipulation and protein production in bacteria, yeast, plant, and filamentous fungi. For the context of our talk today, we see that in the alternative protein industry, there's a wide demand for compelling mimics of animal fats or lipids. One sustainable way to produce such alternative lipids is through microbial fermentation of food waste. Dr. Sofio achieves this using a combination of adaptive evolution, metabolic engineering, and process optimization. In her talk, she will also share her efforts to obtain designer lipid products by altering the substrate specifics of enzymes in lipid biosynthesis pathways. Before I hand it over to Dr. Sofio, I'll just be making a quick announcement of how this seminar will run. So today's seminar will run for a total of 45 minutes with 15 minutes slot towards the end for Q&A. So please prepare some questions as well. We would love to hear from all of you. And regarding that, on the bottom of the screen, you will see the chat and Q&A buttons. We encourage you to use the chat function to introduce yourselves to other participants, get connected. However, please make sure to direct your questions to Dr. Sofio to the Q&A window so that we can address them easily. In the Q&A section, you will also have the option to upvote questions you would like to be answered so that we can prioritize those that are most relevant to the audience. With that, I'll give the stage to Dr. Sofio. Please take it away. Thank you.
Hello, good morning, everyone. Please give me a second to share my slides with you. So I guess everyone can see my slides. Okay, so good morning, everyone. I'm Nazneen, and thanks, Aaron, for the kind introduction. Today, I'll be talking about my work here at SIPI, where I'm trying to produce alternative lipids, or trying to change the lipids production through different techniques like adaptive evolution, metabolic engineering, precision fermentation, and protein engineering. So going ahead, why do we need alternative lipids? Currently, food oils and fats that we use in our food are generally from animal source or plant source. So, but then it's not sustainable in the long run because of two reasons. First of all, the plants that we grow in agricultural land, there is limited availability of the land, and we can't increase the agricultural land without destroying biodiversity further. And as we all know, there is a huge impact environment-wise because of the animal rearing industries. The animal rearing industry produces a huge amount of greenhouse gases. So, and also what we see currently is that the alternative meat industry that's upcoming, we need some sort of animal fat to enhance the taste and flavor of these alternative meat that we are producing. Currently, we generally use plant oils like coconut oil or palm oil, but that does not really mimic the actual animal fat taste. So there is definitely enhancement required in those terms. And there's another thing about increase in the animal fat need. So we need there's an expected increase of about 10% of animal fat consumption over this decade. And how do we cater to that without destroying our environment further? So we do need an alternative production, and most importantly, probably microbial production, where we can use precision fermentation and have less stress on the environment. So I'm talking about this custom lipids, animal lipids. Why are they important for food? So think about you're having a chocolate, and the chocolate is just melting in your mouth, right? It's amazing. How does that happen? Because the fatty acid composition of the chocolate is what determines its melting point. That melting point is just around the body temperature. So when you just have that chocolate, it just melts in your mouth. So that enhances the flavor or the experience of the food. Similar with ice cream or pastry, the lightness that you experience is all because of the fatty acid. So the melting point, the crystallization property of this fatty acids really determine the experience you have as a food. So what does this fat consist of? This fat consists of this tag molecule here, which is a glycerol backbone and has 3 different fatty acids at the 3 different positions of the tag molecule. And the saturation level or the chain length of these fatty acids determine the properties that I just talked about. Say, for example, you have an animal fat, which is mostly solid because it has saturated fat predominantly, and these saturated fat can stack up or arrange itself on top of each other, just so the melting point of the fat becomes higher. Whereas for a plant oil, we generally see it's liquid, like olive oil or palm oil, not exactly palm oil, but olive oil, whatever mustard oil that you use in the food, is because there's a high amount of unsaturation in those oils. So the unsaturation creates a bend in the structure. These fats cannot stack up so well, and hence the melting point becomes lower. So somehow these properties really affect the food that we experience, and hence it's important to have these properties incorporated into alternative meat or whatever we are trying to achieve. So let's dig a bit deeper into how this animal fat looks like. So if we analyze the total fatty acid composition of these tags that I talked about, we can see that this is the data from literature. So there has been a lot of analysis already out there where you can see how the animal fat composition is. So you can see for beef and sheep, the C16 and the C18 zero. So C16 zero meaning you have 16 carbon long chain with no double bonds, and then C16 one would be 16 carbon long with one double bond in there. So you can see for beef and sheep, very distinctly, the C18 zero is quite high compared to the chicken, whereas the C18 2, which is more unsaturated. So you can see that for the chicken, the unsaturation is quite higher compared to beef and sheep. And we all, if we have tasted this food, we know how it feels different when you eat this meat. So probably the fat has something to contribute to this experience. So what we did was we looked into the literature and analyzed all the compositions that have been reported. Also, our team, they actually did buy the meat, analyze the fat in them. And from there, we came up with a target fat composition that we want to achieve. Say, for example, here, this is the target fat composition of the chicken that we are trying to achieve. I'm not giving out the exact composition yet that we're targeting because this is all a bit unpublished. So you can see that this is the kind of composition we will be trying to achieve in our project. So what are the big objectives that we're trying to address here? First is, how do we produce alternative lipids sustainably by probably having some positive environmental impact? And how do we tailor the composition of the lipid basically? So the first part, what we have done is we have secured a food waste stream. We are growing microbes on this food waste stream so that we can improve the lipid production from these microbes. And secondly, for the second part of the talk, where we are tailoring the tag composition using different genetic engineering and metabolic engineering tools. So for our work, we are using microbes, as I mentioned. What kind of microbes are we using? We are using oleaginous yeast. So these yeast are very special because they're not like the normal yeast. They produce a high amounts of lipid naturally inside of them. So what happens is we're just trying to hack that machinery and make it produce something that we want it to. For example, you can see these are a list of different oleaginous yeast that's available that people have been studying. And you can see the lipid percentage of its whole biomass is quite high. Some of them are like 60%, more than 60% lipids. So you can imagine if we produce these organisms, it's going to enhance our lipid production considerably. So how does these yeast metabolize the lipids? So for example, you're feeding a carbon source like glucose to this organism. Through different pathways, it produces this molecule called acetyl-CoA. It's a 2-carbon molecule. And then this goes into fatty acid biosynthesis pathway, making different fatty acids. Generally, fatty acids are made as fatty acyl-CoA molecules. These fatty acyl-CoA molecules are then transported into the endoplasmic reticulum, where they're either converted to longer chain carbons or they are desaturated, introducing different double bonds. So from C16 can go up to C18, C18-1, C18-2. So this creates a pool of fatty acids that can then go into the tag molecules that I talked about. So these fatty acids then are added sequentially to the 3 different OH groups of the glycerol backbone, thus producing triacylglycerol. This is the major component of any animal fat, the tag. And that is what we are interested in. So this pathway is what we focus on in our work. And the tag molecules are then incorporated into lipid bodies, which we can extract out of this oleaginous yeast and analyze to see what kind of lipids these organisms are producing. So to give you an example, this is not totally new. A lot of people have created different kinds of lipids using this organism. So for example, this is a very recent paper which came out last year, where they're making cocoa butter substitute through metabolic engineering of Yerevia lipolytica. So I talked about this pathway in my earlier slide. So you can see from acetyl-CoA through fatty acid biosynthesis, you can get different fatty acids. So what they did was they changed this gene called OLE1 from a different organism. So they completely eliminated C16-1, and now the new engineered strain was producing only C18-1, and cocoa butter has no C18-2. So they deleted this gene FAD2 to do that. So you can see here in the first step, when they are moving the OLE1 to a different one, there's complete elimination of C16-1, which is indicated by the light blue. So you can see there's no light blue bars here, which means there's no C16-1. And once they eliminated the FAD2 here, which is the second step, they could almost see no C18-2. So there's no C18-2 in here. So you can see that by tuning these metabolic pathways and changing some key genes in the pathway, you can really produce some products of interest. So yeah. So what are we doing is we are using, actually working on different projects, and we are using different approaches in these projects. So first of them is adaptive lab evolution, where we have secured a cheap and sustainable carbon source and through waste valorization. And we are growing our oleaginous yeast on these strains and on this carbon source, and trying to get some different kind of lipids. We are also using metabolic engineering techniques where we can tune this pathway, as I mentioned in my previous slide, to get some target lipid profiles. What we can also do is enzyme engineering, where we change the enzyme itself, do some tuning in there, change some amino acids so we can get some target profile, like different products from that protein. Today, my talk, I'll be focusing more on this first and third part in the interest of time. So going ahead with the first part, I would like to mention that this first part is actually a GFI funded project that I was awarded last year, and we have been working on it since last April. So I'll share what we have been doing in this project and some initial results, some key results on that. So how can we improve lipid production using adaptive evolution? So we had this waste stream, and we grow the cells first, the wild type cells, strains that we have. Then we gradually take this and put it into the food waste stream in increasing concentration. So the idea is, if you keep exposing the cells to certain concentration and increasing certain concentrations of the waste stream, it will start adopting and grow in this waste media. So once we have done the evolution, we have some candidate strains that is growing better. Then we do a high throughput lipid screening using stains like 9-red. So we can screen for better lipid producing strains. Once we have those candidate strains, this is a bit high throughput. Once we cherry pick our strains, then we do GC analysis, gas chromatography analysis, which is a more accurate analysis, and it also tells us what the fatty acid composition of those lipids are. And once we have that data, we can do two different things. We can put them into a bioreactor, do fermentation, and see what kind of lipids they produce. So in our projects, we have gone up to about 5-liter bioreactors. And what other thing we can do is we can also do targeted strain engineering to fine-tune those lipid compositions to exactly what we want them to be. This is the overall flow of the project. Now I'll talk step by step, a bit more detail. So as I mentioned, we need to do evolution first. So I talked about these different kinds of oleaginous yeast strains in a previous slide. So we picked two different species. One is Yerovia lipolytica, one is Rhodotorula. And then we had some different strains of these species, and then we started testing them on the waste media. So this first graph here you see is without any carbon source. This is with free fatty acids. And then once we start putting in increasing amounts of the waste media, which is actually very carbon rich for us. So you can see the cells grow a bit better until 10 grams per liter. And then there's a huge decrease in 20 grams per liter. So these are waste streams, right? So we really, though we know exactly, we can find out what is in it, but we still can't be sure of different kind of inhibitors that could be in there. So definitely it's stressful to the wild type, and that's why we need the adaptive evolution so that we can grow them on higher concentrations of the waste stream. What we also did at this point was we analyzed the fatty acid composition of these wild type strains. So with these two information in hand, like how it's growing, what concentrations it can tolerate, how much we need to improve, and what is the fatty acid composition and how we need to tweak it, we went ahead. So talking about the evolution of the oleaginous strains, we took the strains, grew them in a standard lab media first, YPD, and then we exposed them to about 10 or 30 seconds UV, and some were not even exposed to UV, they were just as is. Then we started putting them in increasing concentration of the food waste, like 10 grams, 20, 30, up to 40 grams per liter. After about 80 passages, we call these transfers as passages, which we could then started to analyze what's happening to these strains. So we do analyze the strains after each passage, see how they are behaving, and then go ahead. After the passages were finished, we need to check whether these evolved phenotypes that we are seeing from this strain are actually stable. So we put them back into a standard lab media, the YPD for yeast. So what happens is if your change is not stable, it'll go back to like the wild type. But after we kept them in YPD for a few passages, we introduced them back into the waste stream. And then we saw how these cells are growing in the waste media. So after all this process, what we actually found out was that the rhodotorula toruloids that we were passaging didn't really evolve well, whereas the Garovia lipolytica strains evolved successfully, and I'm going to talk more about that. So as I said, we did the stability assay. So we took the strains, put them into YPD, and started testing the OD. So this is day 1 in YPD, day 2 in YPD, and day 3 in the waste stream. So if you look at this graph here, the blue bars are the ODs that we saw in, or the cell density, you can say, in the YPD. So what we are really interested in seeing is the green bar here, which is how the cells are growing in the food waste stream after we have done the stability test. So you can see compared to the wild type here, some of these evolved populations that we're testing are significantly higher. So we took two of these populations. I say population because once you're evolving a culture or a media, it has a lot of different colonies, right? And all of them would not be evolving at the same pace. So what we are observing is a collective. So we really need to isolate these single star performers that have really evolved well in the waste stream and then analyze that. So for that, we need to do the single colony isolation. So we picked these 2 well-performing strains and went ahead with single colony isolation. So what do you do in a single colony isolation? You first take this evolved population, grow them for a few hours in the waste media, and then you take this media that's been growing for a few hours. So the idea is, say it's been growing just for a few hours. So if your cells have really adapted, and if they can really grow well in the waste stream, they would have grown by now. So then you plate it on a YPD plate. So what happens is you select for these fast growing strains that you have basically. And then from this plate, the next day you screen for about 20 random colonies for good performers. This is basically to enrich the better performance in the population. So I'll show you an example here, one of the populations. This last one here is the wild type. So you can see the 20 random colonies that we selected. Most of them are significantly higher than the wild type, better performers. And also some of them have about a 1.5-fold improvement in terms of OD. So we can say that we have successfully isolated some colonies that are performing, that are growing better in the food waste stream. I would like to mention one thing here is that the OD is not really the biomass. A smaller improvement in the OD itself can really translate into a lot higher biomass, which I'll talk about later. So we selected some of these strains, single colonies, and then went ahead to characterize them. So if you take these yeast cells, you can actually add some dye to it, Nile red in our case, and this will light up all the lipids inside the cell. Then you can look at them under the microscope. So here you can see this is just normal fluorescence microscopy and not confocal. So you can see that the wild type here has some lipids, whereas our evolved strains have visibly higher biomass or higher number of cells in the culture, and also they have higher lipids. Well, this is all very visual. You can also quantify them. So you can take the yeast cells, put the Nile red, put them into a 96 well fluorescence plate, and read it in a plate reader. So it's more high throughput, it's quantifiable. So as you can see, some of these strains have quite higher lipids, almost 10% higher lipids compared to the wild type. So at this point, we can say that we have a better growing strain. We have a strain that has higher amount of lipids compared to the wild type. So we need to analyze them more. So before we go there, what we also did was we optimized not just the food, the evolution work, we also optimized some culture growth conditions and bioreactor run parameters. As I mentioned, this work is unpublished, so I'm not really talking about what conditions, but you can see here that this is lipid compositions in evolved strains using GC, gas chromatography. So this is more accurate, not like it's very quantified data. So you can see that in the condition 2, our biomass has more than twofold improvement, whereas the total lipids also for this condition 2 is greater than twofold. So we can see that by a combination of evolution, by combination of tuning the culture growth conditions, the bioreactor run parameters, we could really increase the lipid production in these oleaginous yeast. And then we also analyzed lipid production for all the strains we had generated. So from some of our candidate strains and growth conditions, they match, they are very close to the fatty acid composition of the chicken fat that we had targeted. Yeah, so really glad to share that. So at this point, to understand further the molecular mechanisms that are key genes responsible for this change that we are observing, we are actually doing a whole transcriptome analysis to RNA-seq. Probably at the next presentation, I can share more about this. So to summarize this part of my talk, we have been growing oleaginous yeast or microbes on food waste streams and producing some tags or tricyclicerols of interest. So we are almost mimicking certain animal fat, and also we are. So by the use of this cheap and sustainable carbon source, we are doing waste valorization from the food industry. We can say that this is an ethically sourced animal lipid that we are producing. And also we are striving to create some positive environmental impact through this work. So moving on to the next part of the talk, it's enzyme engineering, where we are doing targeted lipid changes through protein engineering. So I have been talking about this part of the pathway where we are making different kind of fatty acyl CoAs through our previous work. Now I'll talk more specifically on this part of the pathway where we add different fatty acids to the glycerol backbone chain. So why is it important? As I say, different lipids have different composition. For example, if you see human milk fat, it has this kind of tag. So O is oleic acid, C18-0, P is palmitic, C16-0. So it's different fatty acids at the 3 different positions of the tag molecule. So for human milk fat, we can it has been found out that there's C16-0 at SM2 position very specifically, which doesn't happen in plants or yeast. So it's very specific to human milk fat. Also, palm oil has a very specific tag profile where you can see higher amounts of C16-0 at the first position here, and then C18-1 at the SN2 positions. Also, BFAT has majorly C16-0 at first position and C18-0 at SN2 position. So these are all published data. This is from the literature. So we can see that these different fats have very different kind of fatty acid at certain positions. And probably this is what contributes to its texture or property or melting point, crystallizability, etc. So it's very important that we get the specific tag compositions for when we are trying to mimic animal fat. And how can we do that? So this is the 3 different proteins that add these 3 different fatty acids. So this is a glycerol backbone. The GPAT enzyme adds at the first, then the second enzyme adds here, second position, and then the third enzyme DGAT adds at the third position. So basically, if you can do enzyme engineering of these 3 different proteins, you can get a new tag. You can change enzyme 1 to change this fatty acid and so forth. So how can we use protein engineering? So I'll give you an example of a previous work that I have done. So here, this is not about the tag. So this example I'll give you is about the fatty acid biosynthesis pathway itself, where the acetyl-CoA is added. It's the 2 carbon that goes into this cycle, and then you repetitively add 2 carbons to produce longer chains. This is a work I did as part of my PhD. So what happens is you have this starter molecule acetyl-CoA that goes into the fatty acid biosynthesis pathway. So if you can change this starter molecule here to a different kind of, not just 2 carbon, but different longer chains, then you can ultimately change the product of the pathway, right? Just by changing 1 enzyme. So that's what I did in this project. I did an enzyme engineering of the acetyl-CoA synthetase that produces the starter molecule so that we can get some designer fatty acids at the end. So to do any kind of protein engineering, the first thing you really need is the structural information. How's the protein structure? So luckily for this enzyme, we had many structures from different organisms already solved. And based on this information, we could figure out what's the binding pocket of the substrate. And I was working with the plant enzyme itself. So I determined the identity and the similarity of the plant enzyme to the bacterial enzyme, so structure had been solved. And we saw that the identity is quite high, so we could really use the structures that have been reported in the literature already. What I also did was I analyzed the sequences of similar enzymes, but using different kind of substrate, like 3 carbon, 4 carbon branch chains, and saw how the key amino acids differ in these kind of, this family of proteins. I also did a multiple sequence alignment of all these structures and identified some key amino acids that are changed when the substrate specificity is changed. What we did next was to generate a structural model and analyze the binding pocket. So you can see here, this is the substrate binding pocket, and there's 4 amino acids that line this up. So we can play around with the composition of these 4 amino acids and change the substrate specificity. So what we first did was we changed the valine to an alanine and tryptophan to a glycine, and we generated some double mutants. And we could see that this double mutant here, indicated in red, doesn't use the 2 carbon long chain at all, which is acetate. It actually utilizes butyrate, which is 4 carbon long. So we just by changing 2 amino acids, we could really change the substrate specificity to a longer carbon chain. Using this information, we designed some triple mutants of the same binding pocket. And as we can see here in this images, that some of this binding pocket compared to the wild type here is quite huge. So this triple mutant here, as mentioned, when we analyze its enzyme activity, we saw that it doesn't use 2 carbon at all. It uses 5 carbon, 6 carbon, 7 carbon long chains very efficiently. And also, interestingly, it uses some of these branch chain substrates like 4-methyl valerate, which is a 4-methyl group at a 5-carbon long chain. So it's what I'm trying to really say here is that just by changing a few amino acids, you can really change the substrate specificity and make fatty acids that are very designer. So based on, this is a rational mutagenesis that I did based on structural information available and some initial mutation experiments. So we can see that just by, this is a technique that we can use to actually then change our tags. And these are the 3 enzymes that's been identified. And we have this information. There are a few structures available. Currently, this is a different project that we are doing. It's in very early stages right now. And what we are doing is we're doing the modeling of the enzymes that are involved in the tag biosynthesis pathway. We are also using machine learning because now we can to predict some enzyme designs, some novel ones. And then we will be expressing these enzymes in yeast, this oleaginous yeast, and analyze what kind of tags are being produced. So basically, using the work or the experience that we already have, we are trying to put it into a different pathway and figure out what to do. And figure out what kind of changes we're going to make to these to these proteins and probably get a tag of interest. So actually, I come to my summary. First of all, what we have succeeded in doing is adapting and evolving standard yeast strains to grow in food-based media. We are getting higher lipid amounts and better growth in these evolved strains. We have some promising candidates that mimic food oils and fats, for example, chicken fat. We are also doing culture conditions, optimizing culture conditions to get some desired lipid profile. We can also successfully alter enzyme specificity using protein engineering to get desired lipid products. And of course, the most important slide of the day is all the acknowledgements. I would really like to thank Aaron, Eric, Christian, and Dr. Prakash for his inputs. Aaron and Colin especially for all the GC analysis they have been doing. Christian, Eric, Benedict, and Grace for the fermentation work. And of course, Jingyang, Minjin for their amazing work on the adaptive evolution. So yeah, and of course, GFI for funding this project. And thank you to my audience for having patience and listening to my work. I'm really grateful to GFI also for this opportunity to share my work. Thank you, everyone.
Thank you so much, Dr. Sufio. That was a really wonderful talk and really great to hear you explain your research process and what are some of like the target outcomes and applications in all of this lipid-based work that you're doing. So yeah, I really appreciate that a lot. And I really enjoyed your talk. Let's now go into a Q&A session. And I see that we already have a couple questions coming in to the Q&A box in the Zoom. So we'll start there. And then for the rest of the audience, please feel free to continue adding questions. And we're going to have roughly 20 minutes for this portion of the seminar now. But yeah, for the first question we have is, I guess this is from analytics point of view. What is the absorbance wavelengths for NAL-RED dye spectroscopy? Yeah.
So for the absorbance, there are 2 different wavelengths. One is around, sorry, I don't remember it, but it's around 530. So you can actually see if it's a you can also determine if it's a phospholipid or an actual lipid body, neutral lipid basically. So there are 2 different wavelengths at which I don't remember that quite at this point of time, but I can get back to you on that. Yeah, I just have to see what it is.
Yeah, actually, I have an add-on question to that. In the first portion of your talk, you were discussing high throughput screening, and then tagging some of the microbes beforehand. Could you provide a little bit more detail about how that works? So it's a gene tag, and then you're looking for fluorescence, and then You do like an
Oh,
LC-MS-GC for fatty acid analysis, or?
so we actually do Nile red staining. So it's very
Ah.
easy. So you take your cells, you add spike in Nile red and measure it in a plate reader in a high throughput manner. So it's just a very quick assay. And then you can quantify the amount of absorbance you're seeing compared to your blank, compared to your wild type. And that's when you can figure out which ones to pick for the next step, which is probably GC and also some microscopy work. Yeah, it's quite simple actually. Yeah.
Okay, yeah, good to know. Thanks. Yeah, and then our second question we have here is, yeah, I was quite interested in this as well. What kind of food waste streams do you use?
Again, as this is quite unpublished, but I can say it's a very carbon-rich source. So we can, and there's some sort of other compounds in there, but probably you can have to wait a few more months for it to publish. We are currently writing it up, so we can share more at that point. Yeah, but it's a carbon-rich source. It's a solid waste actually. So Eric and Christian in the team, they actually developed a way to kind of dissolve it into a way, in a way we can actually grow our cells on that. So yeah, that's also a hurdle that is there. So it's very solid waste. Yeah.
And just to understand like a little bit more about the development time on optimizing these food waste streams, how long did that work? Like how long did it, I know you can't speak to the identity, but like what was the process in terms of like identifying, did you guys look at different options and then like screen some and then kind of like work on a couple?
So actually, so what happened was we were working on a different project and we had this, had the accessibility to this kind of food processing waste. And then so the members in the team, they came up with the idea of utilizing. So we tried first just because we knew this is the composition. We just were interested because we had our, like we actually got that food waste stream from a different project. But then we also have a team at SIGB doing this, the circular bioeconomy team led by Christian, where we have access to a wide variety of food waste, but not for this project, but there's a lot more projects going on at SIGB where we have access to all this food waste and we can grow different kind of microbes on this food waste to produce different kind of products. So yeah, we do have a team here at SIGB that's working on it. So yeah,
great.
That's how we have access to this waste. Yeah.
Great to know. Thank you so much. And our next question is, what is the potential of scaling up this process?
So we have actually done 5-liter bioreactors already. So the only problem that we see is that we can only go up to 20 grams per liter of the waste in terms of bioreactor, because there's a lot of logistical issues like foaming, etc. that's happening. So so we have tried up to 5-liter bioreactors that we can at this point. We haven't tried more than that. So yeah, that's something we need to look into the future in the future, like how can we potentially scale it up? Yeah.
Right. And kind of alongside the scaling up, in terms of getting to the product application as well, like is there a purification or separation step for like extracting these liquids out of the microbes? And is that something your team works on as well?
So actually, that's definitely on our minds. Like, so the organism we are using here, Ovia, is actually grass status.
Great.
So, but of course, we right now do extractions for GC anyway, but it's small scale and we use organic solvents. But we have talked to some companies regarding this, and I think industry is open to using some food grade organic solvents, but we do have to extract it out. So, but then this is not a in the scope of the present work that I'm doing, but this is definitely future work that we want to delve into. Yes.
Yeah, especially like for food applications, things like GC or maybe like column separation can get quite expensive really fast. And
in terms of that, I think it would not be so much, because as I said, the biomass, the lipid percentages of the biomass of these organisms is very high. So sometimes also you can see the lipids like floating, the cells floating up if they have too much lipids. So I think the downstream is not going to be that difficult. It's going to be some simple extraction process. Yeah.
Awesome. Great to know. Thanks. We have a couple questions coming in, so I'm going to try to get to those as well. Our next one from Shital Parak is, how do you decide on the specific number of passages required for sufficient transformation during ADE?
Oh, yeah. So basically what we do is we take our cells, we grow it for one passage, we observe what's the OD. So we go on continuously, and after a certain number of passages, what we do is we do an analysis of all the population with the wild type and see that, okay, how much it has improved compared to wild types. And then at that point, we say, okay, we take these two and then proceed further. So then passage it more. So basically, it's a trial and error in the sense of like, you have to analyze at certain time like breaks, like if you have achieved enough. Also, sometimes what happens is you see a contamination happening, so then you have to trace back. So you have to make like glycerol stocks of every day that you passage. And so you go back to a certain generation again and you restart from there because you have figured out there's no contamination. Because this is growing in a food-based stream, sometimes contaminations do creep in. So you have to go back and forth and you just have to rationally decide what's a good point. I think you can, so 79 passages should be around, like if you calculate it, it should be more than 200 generations. So that's a good number. If you have actually had 200 generations, then generally it adapts well. But again, it depends on the species, as Rhodotorula never evolved for us, whereas Yarovia evolved for us. So it depends on the species also. You just have to check along the way, I guess, what's good enough for you. Yeah.
I see. Cool. Good to know. And our next question here is, what are some red tapes behind this project? And the person who asked the question said that if I understand correctly, the workhorses are genetically modified. Does it comply with current food safety regulations? I believe you addressed this a little bit as well, but if you could go into more detail.
Yeah, so basically the strains that we're generating using adaptive evolution would not be considered genetically modified because we are just naturally evolving the strains on the waste. We are not doing any modification on that. So whatever fatty acids that I said that we have some candidate strains where we did the evolution and we changed some culture conditions, is zero genetic modification. I actually did not talk about the metabolic engineering part in this, in the first part actually. So those can go into, so that's the idea. We didn't dwell too much into genetic engineering in the first part, so we could get some food safe strains. But for the second part, yes, it would be genetically modified. But then what my understanding is as of now, that if you can remove all the genetic materials, all the organisms from your product eventually, it could be tagged as non-GMO. Maybe my understanding is not enough. I'm just a scientist, but yeah, maybe other people in the audience can comment on that more if they know. Yeah, but that's my understanding.
And I guess with the metabolic engineering and enzyme engineering aspects, you would probably get into scenarios where it could be considered GMO and non-GMO. But I think what really matters too is it depends on like the target lipid you're making and whether you purify and separate it. Because if none of the GM product is in the final outcome, then I think there's considerations there very specifically about whether it's GM or not GM. But yeah, that's I think that's a very long discussion within itself. Yeah.
But for the first part, it's not GMO as of now. Yeah.
Right. Our next question is they mentioned thanks for the amazing talk and great work, as well as could you please elaborate about end product formulation? Do you isolate the fatty acids as end product? And I think just kind of plugging into other questions as well is like where do you see the application? Is this to go into a burger patty or a chicken breast, I guess, after that whole like separation aspect has happened? Yeah.
So as of now, for our scientific analysis, we do isolate the neutral lipids, the tags basically, because we need to find out there could be free fatty acids also. We need to figure out what's attached to the tag molecules. So we actually isolate the tags and then we cleave off the fatty acids from there but that's just very technical in terms of analyzing the composition. But as a product, I think again it comes back to the question of how do we extract it, and once we extract it, then how do we use it in an alternative mean? So honestly, that's not something we have come to that point yet in the project. We are still working on tweaking the lipid composition. So yeah, that's definitely something. VCB does have the food processing team. So hopefully in the future we can work on that aspect as well. Yeah.
Right. And I guess that will come along with like a certain level of scaling up as well, because you need to generate enough mass to be able to actually like do the more so like food science, material science aspect of incorporating it into formulations and stuff.
Yes, yes.
But yeah, so maybe not at that stage quite yet.
No, I wouldn't say so.
Yeah. And our next question is from Nestor. How convenient would it be to implement a similar pipeline of research in algae, considering that these microorganisms need inorganic carbon sources rather than sugars, and algae are quite capable of producing relevant amounts of lipids? And just for me to add on as well, like how about other microorganisms as well, like Pichia or E. coli or things like that?
Yeah, so basically I think in terms of lipids, we chose oleaginous because they make a lot of lipids, but in terms of that, algae could also be a good candidate. I have no experience working with algae, but I think if we try to adapt algae, it would perform quite well, I guess, because there's a lot of literature available already on adaptation of algae. In terms of E. coli and Pichia is again grass, I think, so and also a yeast. But the amount of lipids being produced from these organisms are not as high as oleaginous yeast. So I would any day prefer some sort of organisms that naturally produce a lot of lipids, so we don't have to do a lot of metabolic engineering on it to increase the amount of lipids. I think to answer this specific question, I think it would be okay to also work with algae in this kind of a pipeline. Yes, I would say yes. Yeah.
Right. So definitely options for other microbes as well.
Yes. Yes.
And then our next question is, how do you test the bioavailability of these fatty acids?
Yeah, I think that would be, basically we are already eating tags. So if we are talking about bioavailability when we consume these fatty acids, I think it depends, as I said, on the extraction. Again, if you just take out my understanding would be if you just take out the tag from your microbe and then just incorporate it into your meat product or something, it will be as available as any other fat in the market right now, because it's a tag molecule, and we are not making anything new. We are just making the things that's already in naturally, like C16, C18, C18-1, and C18-2. Those are the major components. So we are not making anything new. So my idea would be the availability would be same as the natural fatty acids that we eat. We're just mimicking it. We are not making anything completely novel or anything, in these. So, yeah.
Great. And I think maybe we'll have time for roughly 2 more questions before we do some closing remarks. But we have our next question from Nestor, which says, what is your opinion about the possibility of designing this biological system to be fed with simple 1-carbon precursors, such as formate, small alcohols, and so on?
I think there's already some work in the literature where people are growing this yeast on single carbon sources. So I think there's definitely a work that's ongoing already by different teams in the world where they are working on single carbon sources. So yeah, I think there's definitely possibility of it. Yeah.
Right. But like the focus on your team would be like
Waste valorization.
Yeah.
Yes, currently. Yeah.
And are just curious, I know you guys are working on that one waste stream right now. Are you also in considerations of incorporating, like exploring other options as well, or staying focused on this one waste stream?
No, we are actually also trying working on potential projects where we could actually work on different kind of waste, not just this one. Something that's even more logistically easier to scale up, etc. Yeah, definitely. But using the learning from this project and going ahead. Yeah.
Great. And yeah, let's maybe for one last question, or if anybody else has any burning questions, like please feel free to add it to the Q&A box right now, and we'll see if we can address that in time. But from Sandra, we have a question stating that what is the proportion of the fatty acids attached to the TGs that is produced de novo?
De novo, yeah. Yeah, this is an interesting question. Yeah, we have actually looked into it. So basically, as I said, we changed the culture condition, right? So what we see is that if we just give it more and more food waste, it starts accumulating because these cells can actually take up this carbon, and then it is. I think. I'm not sure if I understand. So basically, so these cells take up this carbon source and then produce these fatty acids, and that is attached to the tags. We are not actually analyzing the fatty acids that's free in the cell because we are more interested in the tag itself and what kind of fatty acids are actually attached to the tag itself. Does that answer your question, Sandra?
Yeah, Sandra, please feel free to follow up as well if
not, you can always send me
a
follow up later.
Yeah, and I will be I'll be putting Dr. Sophia's email in the chat shortly too. So yeah. But yeah, actually let me do that right now so that anybody here can feel free to reach out to her with any further questions, or if you're interested in her work further. Yeah, and then looks like we have maybe time for one last question. Is how do you compare the microbial bioprocessing to the mammalian adipocytes bioprocessing to produce the fatty acids? So microbial versus mammalian adipocytes.
Yeah, so yeah, so basically the pathway is quite conserved. There are homologous enzymes in human and microbes. So the tag biosynthesis pathway that let me pull it out. So basically the tag biosynthesis pathway that I mentioned in the slide here, the different enzymes are actually conserved. You can find similar enzymes in humans, similar enzymes in microbes. So these kind of very pathways are quite conserved. So I think, yeah, it's quite similar. Maybe the sequences are different of these enzymes, but they are same family of enzymes. So yeah.
Similar pathways. And then, yeah, just maybe the comment from Sandra for the question mentioned before was that she said, I posted the question because you're feeding the cells with waste and it's possible that part of the fats can come from this source.
Oh, yeah. Okay. Okay. That makes the question quite clear. Yes, definitely the fats can come from this source. So what we have observed is we change the culture conditions, we change the growth conditions in terms of not the composition of the feed, but other parameters. And we have seen that we can tweak the profile quite a lot. which says that it's not only, definitely there are fats in the waste media that the cell is feeding on, but also the cells can then put this into their, utilize it into their pathway and create different products. So yeah, we have definitely seen that. And also, if you grow it for a longer time, if you increase the amount of time, you can see the change in the lipid composition slightly, which means that it's actually, if there's any fat in there is some fat in the source also, not so much, but then it is actually getting even changed in terms of the product. So yeah.
But yeah, with that, I'd like to, well, firstly, thank the audience for all the great questions. And thank you, Dr. Sophia, for her answers. But also, yeah, I just want to say thank you for your insightful talk today. And thank you, everybody, for attending and engaging. And we'll see you in a few months' time for our next seminar. So stay tuned. And to everybody, thank you so much and have a great day.
Thank you.